So the cells of the nervous system, we have two major cell categories, the neurons and the neuroglia, or the neuroglial, or the glial cells. They're all the same thing. It's just different names for the same thing. But they have two very different functions. So the neurons, that's this cell right here. You've seen this in lab. You've seen it when we went through the tissues. It kind of looks like a really weird palm tree. Neurons have two jobs. I receive a signal and I transmit it as an action potential. Those are my two jobs. I get info and I send it along. I'm a relay center, okay? I get my message and then I pass that message along. Anatomically, at the top here, this big chunk up here is actually called the cell body, okay? The neuron cell body. It can also be called the soma, just so you're aware, okay? S-O-M-A, which just doesn't look very well written there, but that's what it can also be called. This is the primary site of protein synthesis. This is where you make protein. You may transport that protein all over the place, but this is where I make most of the protein in my cell. Internally, I've got a nucleus and I've got a little teeny tiny body in there that's dense called the nucleolus. I also have, just like in regular cells, the Golgi, the mitochondria. Now, something that is named a bit different, but actually is something you already know, the Nissl bodies. Nissl bodies are the primary site of protein synthesis. It's the rough endoplasmic reticulum. But remember what I told you about scientists and naming things that have already been named into something else just because I can? Yeah. Nissl bodies are going to be your rough endoplasmic reticulum. It's the primary site of protein synthesis. But because it's in a neuron, we have to name it something different because we're scientists and we have to be complicated. If you look, here's my cell body. But do you see all of these other kind of short projections coming off of it? In fact, if I were to cut this off and just show you from here up like this, kind of looks like a tree with branches, right? These are called dendrites, okay? Dendrites are actually extensions of that cell body, but they're very short. And coming off of a neuron cell body, you usually have a lot of them. When we talk about dendrites just in general bio, we talk about the fact that they're kind of the thing that picks up a signal. Okay, they're the ones for incoming info, which kind of isn't true, but we'll talk about that in a bit. Okay, then down here, I have one huge, large extension coming off of the cell body. There is only one axon per cell body. And it's always the longest. And you can always tell it's the axon because it is the longest. If I point to anything else and it's short, automatically it's a dendrite. Okay? So the axon carries signal away. Axon away. Okay? You've got one cytoplasmic extension of the cell body that transmits the action potential from this cell to the other. Remember I said I receive info and then I send it along. This is the sending along. On this, you have the axon hillock. See how this kind of comes down like an ice cream cone? That's the axon hillock. And then I've got the initial segment. Initial means the first, right? So this is the first segment right here of my axon. Together, these two areas make something called the trigger zone. Remember I said that there's a reason I say dendrites are for receiving, but it's not really all that accurate. The trigger zone is the most sensitive part of this neuron. If you hit the trigger zone with an action potential, it is going to reach threshold and go. Okay? It is literally the most sensitive part of this neuron. The axolemma. I'm going to switch back to yellow. The axolemma. Remember that the cell membrane of the muscle cell was called the sarcolemma? Well, the cell membrane of the axon is called the axolemma. Why? Because we have to be complicated. We're just bougie like that. I don't know. Inside, I've got the axoplasm. It's the same thing as the cytoplasm, but because it's in the axon, we call it the axoplasm. So remember that synapse meant where two cells meet. So if there was another cell down here, I love having two completely different colors. If there was another cell down here that this was supposed to be in contact with, these would be the presynaptic terminals. The little feet, the little patitas here, would be the presynaptic terminals. So if you look here, this is what I'm talking about, okay? Basically, it's before that space between the new cells. Now, you already know this because we saw this in chapter 9. Inside of those presynaptic terminals, we have the synaptic vesicles holding neurotransmitter. Okay, this is not new information. This is actually going back to chapter 9. So that you're aware, we're kind of already starting to kind of go through little bits and pieces that you've already seen before. So what we discussed in chapter 9 is exactly what we're discussing down here. The neurotransmitters and the presynaptic terminals, same thing. So the types of neurons. It depends on what they do and how they're built. So I'm going to go with structure first, okay? We're going to talk structure first. If I have a cell body, okay, so here's my cell body, and I have a lot of cell processes coming off of it like this. We actually call this multipolar. Multipolar, because I have a lot of sticks coming off of that cell body. Bipolar, what does bi mean? I'm sure everybody knows it means two, right? So here's my cell body. Here's one process coming off of that cell body. Here's another process coming off of that cell body. So it's bipolar because I've got two processes coming off. Unipolar, which is also now known as pseudo-unipolar. Pseudo meaning fake. Here's my cell body. There's only one projection coming off of it down here that you can see. Okay, so here's my cell body. There's the one projection coming off of it. This is anatomical structural classification. If I'm telling you to look at it and tell me what it is, this is what you would say because of what it looks like. Okay? So, functional classification. Functional classification. You've got sensory, you've got motor, and you've got interneurons. Inter means in between. Okay, so you've got sensory and motor and inter. So I'm going to go back to our original picture here. Whoops, that wasn't what I meant to do. Sensory neurons conduct action potentials from outside in your body up to your central nervous system, bringing information into the central nervous system. Sensory neurons are on this side. Okay? They bring information in to my central nervous system. They conduct action potentials into my central nervous system. So sensory neurons. Do that. Okay, we're doing the exact same thing that we said we were doing before. We're taking information from outside here and taking it up into my central nervous system. Okay? Motor neurons are over here. Okay? Motor neurons. they conduct action potentials from my central nervous system here out into my body. So basically, their function is this function. It's the same function. I'm taking information from my central nervous system out into the body. Again, this is functional classification. This is physiological classification. How does it work? The third function that we have is basically connecting this neuron over to this neuron. See these three little neurons I just drew? Okay. These are the interneurons. Remember, inter means in between. They carry signal, they carry action potentials from one neuron to another. They're the connecting point. They're the ones that actually connect the two neurons together. So, functionally, I have the sensory neuron. The sensory neuron is picking up a signal. I stepped on a nail and it carries it up to my central nervous system, where I get the processing of the information, initiating responses. Do I immediately take action? Do I make a memory? Do I ignore it? And the interneurons are the ones doing all of that work and eventually coming to a decision. Then they send that signal to the motor neuron. The motor neuron's job is to carry that out to the muscles of the body and say, hey, move your foot. You just stepped on a nail, so that I can pick my foot up using my skeletal muscle and basically getting my foot up off of that painful stimulus. Okay? So if you know this, you know function, you know parts of the divisions of the nervous system, and you know functional classification of the neurons. You know all three of these things if you do this. Now, admitted, motor division still has quite a few divisions to it, but for the most part, there's a lot of information just in this one figure. And normally I really do put this up on the board, and normally I really do show you like this, just FYI. Okay, so that's the functional classification of the types of neurons. Now, let's talk about the glial cells, or the neuroglia. These are non-neuronal cells. Remember, I told you they have very specific functions. My neurons carry information. They are the ones that send signal from point A to point B. Neuroglial cells' job is to take care of neurons. They're the babysitters. They're the ones that actually make sure everything is functional so that those neurons that are carrying signal can work the right way. And I have two different types based on location, okay? Or two different categories based on location, I should say. In the central nervous system, I have 4 neuroglial cells. I have astrocytes, ependymal cells, microglia, and oligodendrocytes. Now, central nervous system, you will only find these in either the brain or in the spinal cord. Those are literally the only two places that you have them. In your peripheral nervous system, so not your brain and your spinal cord, you have Schwann cells, apparently also known as neurolemmacytes. I'll be honest, guys, I worked in a neuroscience lab for years. We never called them that. We always called them Schwann cells. And then you've got satellite cells. Okay, so in your central nervous system, you have 4 categories. In your peripheral nervous system, you only have 2. So astrocytes. What's the symbol for this? Houston Astros. It's a star, right? So astrocytes, see this right here? Looks like a starburst. Same here. These are the astrocytes, okay? Don't worry about this for now. I'm going to talk about it, but I don't want you to worry about knowing it for the exam. I'm more interested in you knowing the functions than anything, but I do want to explain this. So I'm going to explain it just so you have an idea of what I'm talking about. If you look at these astrocytes, you'll notice that they've got these little foot processes that are surrounding things, right? They very intentionally do this. One of the reasons is because it's kind of like having the suspension lines on a suspension bridge. Having these kind of stuck around like this helps to suspend things where they need to be. The second reason they cover like this is because they're actually preventing things from just getting out of the blood vessel. So functionally, it's structural support for neurons and blood vessels. Again, it's like I can suspend things where I need them to be. I can put things where I need them to be, and these little processes help to hold them in place. 2, it influences the function of the BBB. BBB stands for blood-brain barrier. The blood-brain barrier basically is a wall that doesn't allow things into or out of your central nervous system. Your brain is one of the most, well, it is pretty much the most important organ in your entire body. Everything else works based off of the function of your brain. So having things just get access isn't really a good thing. So what do? Well, we put up a barrier that makes it very difficult for things to get in. Can things get in? Yes, but you have to have a special pass in order to get in. So these cells, see how they're surrounding that blood vessel? They actually secrete chemicals that promote the formation of tight junctions in the capillary cells. Basically, remember, tight junctions were those junctions between the cells that were so tight nothing could leak between them. If I'm secreting chemicals that are doing that to the cells in this capillary, that means if I want to get out of this capillary into the nervous system, I have to go through the cells, and that's a lot harder to do than just leaking through. They can isolate damaged tissue and limit the spread of inflammation. So my ankle, remember I told you I did something to my ankle and it hurts today, right? My ankle is swollen, but when my ankle swells, there's room for it to swell. There's room for it to spread out because there's space, right? The thing about your brain and your spinal cord, both of them are in a big concrete box. Your brain is in a big concrete box in your skull, and your spinal cord is in a big concrete box in your vertebra. If there is damage to these, they literally have nowhere to go. So if they start to swell up, they're actually going to damage themselves by squishing themselves and damaging against a concrete box. These cells, these astrocytes, actually keep inflammation in one little spot. At least they try to, so that hopefully if they're swelling, it's in that one little teeny tiny area and it doesn't become the whole brain. If your brain does start to swell because damage is that bad, normally nowadays in the present day, they will cut a piece of your skull out and they will either put it inside your body or put it into a refrigerator to kind of save it and give your brain room to swell so that it doesn't do that, hopefully not causing damage. They help to maintain synaptic function. So 4 and 5, 5B, are kind of the same thing, okay? If there isn't enough neurotransmitter, they will create neurotransmitter. If there is too much sodium, it'll absorb the sodium. If there's not enough sodium, it'll release sodium. It keeps the environment that that neuron is in perfect for that neuron to work. That's one of its jobs. Finally, they promote the development of synapse. They will actually promote the development of a neuron touching another neuron. You can actually watch that happen. Neurons kind of making connections. So that's actually really cool. The second type of glial cell in the central nervous system is the ependymal cells. I'm going to read this first and then I'm going to talk about it. Specialized cells that line the ventricles of the brain and the central canal of the spinal cord. Some produce cerebrospinal fluid, and apical surface cilia help to circulate it. Cell base has long processes that extend into the brain and the spinal cord, and in some cases have astrocyte-like function. All right. Ependymal cells have these little cilia up here that beat in rhythm, almost like oars on a boat. If you've ever seen a movie where row, and all of the oars are going at the same time, that's kind of what they're doing. So we have a substitute blood supply inside of our central nervous system. Blood, for all intents and purposes, is very dirty. We don't want blood in there. So we made the substitute called cerebrospinal fluid, right here, cerebrospinal fluid. The problem is that in order for us to have that cerebrospinal fluid, it needs to circulate like our blood does. Well, our blood circulates because we have an organ called the heart. The heart allows pressure that gets our blood to go in circles, that basically allows it to circulate around our body. We don't have a spare heart in our central nervous system to create that pressure to get it to circulate around our brain and our spinal cord. So what do? Well, we have these cells working like oars on a boat, and as they beat together in the same direction, they basically create a current, and that current is how our cerebrospinal fluid actually flows around. Now, I said they're in the ventricles of the brain and the central canal of the spinal cord. Our two major organs in our central nervous system are the brain and the and the spinal cord. We have hollow spaces called ventricles and a central canal in these spaces that actually, that actually have room for this fluid and are lined with these cells that have these cilia that are working like oars to get it to circulate around, okay? They are, to a certain extent, responsible for making the cerebrospinal fluid. So if they're laying on top of your brain and your spinal cord, and in some cases your blood vessels, they're actually pulling the fluid out of the blood and letting it into that space. The thing is, if I have to go through these cells here to get to the other side over here, they get to decide who goes and who doesn't. So them making cerebrospinal fluid actually makes sense. They have astrocyte-like function. Remember I told you, in order for neurons to function, they have to have the right environment. These guys help to make that correct environment, just like our astrocytes did. If there isn't enough, they get more. If there's too much, they take it out, etc., etc. The microglia. They become mobile and phagocytic, phago meaning eat, cyto meaning cell, in response to inflammation. They phagocytize, they eat necrotic tissue, dead tissue, microorganisms, invaders, bacteria, or viruses, or other foreign substances that invade the central nervous system. So remember I said I don't want my blood to be inside of my brain and my spinal cord. This can lead to an issue because my fighting cells are actually in my blood. So if I'm not letting those in, I better have a backup plan. Microglia are my backup plan. Microglia are the ones that when there is something wrong in the brain or the spinal cord, they become our fighters. They become our army to go and do cleanup and to hopefully fight off whatever is in there. Now, the oligodendrocyte. Oligodendrocyte. They've got these cytoplasmic extensions. See how they're extending off of that cell body? And they wrap around axons many times. to form something called a myelin sheath. So imagine that I've got the core of wrapping paper. You know how wrapping paper always comes, well, actually, even simpler, toilet paper roll, right? There's that cardboard roll in the middle, and then you've got the toilet paper wrapping around and around and around it so that you can use the toilet paper, right? This extension basically wraps around this yellow axon like toilet paper does. And it's a lot of layers. Okay, so you've got many, many layers. I don't know why it's not zooming. Nope, not zooming. It wraps around and makes many, many layers of cell membrane on top of these axons. Now, something that's kind of interesting about the oligodendrocyte, you'll notice this one cell is wrapped around multiple axons. That's unique to the oligodendrocyte, okay? When I wrap around a lot, like you see here in this picture, we call it a myelin sheath, okay? So again, the fact that it can do that around several axons is unique to the oligodendrocyte. So in my central nervous system, I have neuroglial cells, including the astrocytes, the ependymal cells, the microglia, and the oligodendrocytes. Now let's talk peripheral nervous system. In other words, not the brain and the spinal cord. We only have two. We've got the Schwann cells and we've got the satellite cells. The Schwann cells are basically like the oligodendrocytes. They wrap around axons and they make that myelin sheath that we were just talking about, where it wraps around over and over and over again. But unlike the oligodendrocytes, these form a myelin sheath only around a portion of an axon. So in that previous picture, you saw that one cell wrapped around 3 different axons, right? Look here. Here's one axon, and I've got 1, 2, 3, 4 Schwann cells wrapped around it. So here in my peripheral nervous system, it's basically, instead of a 1 to 3 relationship, it is now like a multiple to 1 relationship. One axon may have several Schwann cells wrapped around it. The satellite cells. See these cells here that are wrapped around this cell body? You can see the neuron cell body. This is a unipolar cell. You can see the one stick coming off of it. See all of these cells that are wrapped around it? These are the satellite cells. They surround the cell bodies in the sensory ganglia. Now, they do provide them with support physically and nutrition. Remember I said they want to make it the perfect environment for the neuron. So they're there to do that. Something else that is kind of interesting, they protect that cell body from heavy metal poisons like mercury. So basically what I'm saying is, if you were to get mercury poisoning, to prevent it from getting to your neurons, all of these satellite cells would absorb the mercury into themselves and die before letting it touch this neuron. So myelinated and unmyelinated. Myelinated cells come from Schwann cells and oligodendrocytes. They wrap several layers of plasma membrane around the axon of the neuron. It forms a series of tightly wrapped membrane-rich phospholipids with very little cytoplasm between the layers. It gives them a white appearance as well as a high lipid concentration, because what is our cell membrane made out of? Cholesterol and phospholipids. So if I have cell membrane on top of cell membrane, it's going to be a lot of lipids. I want you to look here, okay? I want you to look here. Notice that this is just cell membrane on top of cell membrane. Whoops. It's just cell membrane on top of cell membrane. The cytoplasm is way the heck out here, because this is a living cell. This is a Schwann cell. But where it's wrapped around really tightly, making this myelin sheath, It's just cell membrane on top of cell membrane. The best way I can think to describe it is describing a toothpaste tube. If I want my toothpaste, every single last bit of the toothpaste out of that tube, I'm going to roll it from the bottom. And the hope is that as I roll it, all of the guts, quote unquote, of my toothpaste, all the toothpaste is being squeezed out. And all that's left in the roll that I'm making is the tube on top of the tube, the two layers. That's what this is. It's just cell membrane on top of cell membrane without any of the cell cytoplasm in there. That is the myelin sheath. When I have this in neuronal tissue, it literally looks white, okay? The nodes of Ranvier. So do you see how here there's just a little teeny tiny bald spot, and same here, there's a little teeny tiny exposed region this tunnel kind of creates. These little areas are called nodes of Ranvier. They're going to become important later. Now, this is an example of a myelinated axon. It has the myelin sheaths on it. This, however, is an example of unmyelinated axons. Notice I don't have layers and layers of the cell membrane like I do up here, down here. All I have is that one layer. With that one layer, this is considered unmyelinated, okay? You kind of have these little indentions or culverts that your axons sit in, but they're not wrapped multiple times. Something that's kind of interesting. If I have a myelinated cell, the action potential goes really, really quickly. If I have something that's unmyelinated, it actually goes pretty slowly, which we'll explain.